Welcome! Thank you for watching the cryopreservation of neural stem cells tutorial and also for using our products. Cryopreservation of mouse neural stem cells. Materials required: a centrifuge tube rack, tissue culture flask, a water bath, semi-dry alcohol swabs, a pipette, EP tubes, a centrifuge, 15 ml conical tubes, pipette tips, NCR protein-free cryopreservation medium, a CO2 humidified incubator, neural stem cell growth medium. Cryopreservation procedures. Note: collect cells that are in the logarithmic growth phase. Change spent medium with fresh growth medium 24 hours prior to freezing. Pre-warm the NSC growth medium to 37 degrees Celsius. Note: avoid repeated warming and cooling of the medium. If the entire content is not needed for a single procedure, transfer only the required volume to a sterile secondary container. Observe cells under the microscope. Be careful not to flap the bottom of the culture flask to avoid cell detachment. Transfer the cell suspension containing floating neurospheres to a 15 ml conical tube. Centrifuge at 140 times G for 4 minutes. Aspirate and discard the supernatant. Resuspend the cells in 1 ml of NSC growth medium. Transfer the cell suspension to a 5 ml EP tube. Slowly pipette up and down to mix the cells. Repeat this step 10 to 20 times to obtain a uniform single cell or near single cell suspension. Observe cells under a microscope. For larger culture volumes, aliquot cells into 2 to 3 Eppendorf tubes before pipetting. Note: do not use excessive force when pipetting up and down. Place the EP tube in the tube rack for 2 to 3 minutes. Then collect cells floating in the supernatant. Note: be careful not to aspirate the cell mass in the bottom of the tube. Collect cells that are in the logarithmic growth phase. Perform a cell count to determine the viable cell density. Centrifuge at 250 times G for 5 minutes. Aspirate and discard the supernatant. Resuspend the cells in the neural stem cell NTR protein-free cryopreservation medium at a cell density of 1 × 10 to the 6 cells per ml, or at the desired density. Note: the NCR protein-free cryopreservation medium should be kept at 4 degrees Celsius prior to use. Dispense aliquots of the cell suspension into cryogenic storage vials that are properly labeled. Place the vials directly in the -80 degrees Celsius freezer and store overnight. The next day, transfer the vials to the liquid nitrogen container for long-term storage. Thank you for watching this tutorial.